So yeah, this week we're going to be talking about DNA, and DNA is super cool in the world of crime and solving crimes, because basically DNA is super unique to each individual, and unless you have like an identical twin, no one else is going to have the same DNA as you. So that means that like if your DNA is found, if you find someone's DNA at the crime scene, it's super easy to identify. However, the government doesn't have just like a database full of everyone's DNA. So that's a little bit of an issue. So that's why a lot of other methods are needed to identify like crime suspects, or else DNA could just be all you use, and that'd be really convenient. But unfortunately, it's not that easy. So, but yeah, today we're going to be talking about DNA and just going more into depth about what DNA is and how it works. And then we're going to be talking about how like DNA is used in a lab and how like people see different like how you look at DNA and compare DNA to like different things, and how you can like match DNA samples. So yeah, here's the schedule. We only have 2 more weeks after this, so yeah, hope you guys are liking it so far. Okay, so what is DNA? Most of you have probably heard of DNA before. It is deoxyribonucleic acid, and you can kind of see the D comes from the D, and then nucleic acid is the N and the A. So DNA is kind of like the genetic code for everything in our bodies. So kind of think of it like you're baking something or cooking something, and you're following a recipe. And the recipe is telling you like how much flour to put in, how much butter to put in, how much sugar to put into your cake. But like without that, you would like maybe know what to add, but you wouldn't know how much of it, and your cake probably wouldn't turn out as good unless you're like a professional baker. I don't know. Maybe some of you guys really like baking, but yeah. So that's like the same thing for DNA in our bodies. DNA tells your body kind of like what proteins and what cells that you need to make in your body to make you, or else you might end up like a blob on the ground. I don't know. But that's pretty much the function of DNA. And you guys probably know that this is what DNA looks like. It has this like double helix structure that it like winds around itself. And there's different nucleotide bases, which are these little colored parts on the DNA molecule right here. And so you have A, which is adenine, which always binds to T, which is thymine. So you can see that the purple and the green are always paired together going down here. And then you have the cytosine and guanine, which are G and C and always paired together as well. And then what holds it all together is the phosphate backbone, which we'll go more into depth later, but that's like the yellow part here. And that creates like the spirally winding part. And then if you guys know DNA, you probably also know RNA, which is ribonucleic acid. And the only difference between these two is that they use different sugars. So we'll look at the structure of the sugar in the next slide, but deoxyribose is a type of sugar, and then ribose is a type of sugar, and that's what makes DNA and RNA different. And then RNA is also usually single-stranded. So you can see how DNA has like one on this side, one on this side. RNA will usually just be by itself. And then it has different nucleotide bases. Instead of a thymine, it's going to have a uracil, which is U. So A binds with U and G binds with C. Okay. So this is like more in-depth of the structure of DNA. So this is kind of what we looked at earlier with the double helix and the G and the C binding and the T and the A. But what really does it look like? So this part, if you separate it here, this part is the phosphate backbone, and it's made out of these phosphate ions and these sugar molecules. And the sugar is deoxyribose, which is this thing right here. And then if you guys know, DNA has different ends to it. So this side is the 5' end, and this side is the 3' end. So what it does is it actually runs anti-parallel to each other. So if you'll notice, there's a 5 on this side and a 5 on this side, and the 3 is up here and the 3 is down there. And this kind of shows like which carbon, because on the sugars, these are all of these points right here, which is not labeled, are all carbon molecules. And it binds to the number of the carbon. So the OH here binds to the 3rd carbon, and the phosphate binds to the 5th carbon. So they run opposite each other, and that is what makes like replication of DNA easier. And then nucleotides, here's the structure of the different nucleotides. You can see that guanine and adenine each have 2 rings like that. And then cytosine and thymine each have 1. So the 1 ring is called pyrimidine, and then the 2 rings is called purine. You don't really need to know this, but it's just kind of fun to know. Does anyone have any questions so far? Have you guys learned about DNA before at all? Or like maybe a little bit, or maybe you love DNA, I don't know. Yeah, learn some, okay. So yeah, cool. In school, we're gonna be talking more about like DNA replication and DNA, like you guys probably learned a little bit about mitosis and meiosis, so yeah. And then we'll talk about like different like laboratory techniques. Like, have you guys heard of like PCR and gel electrophoresis before? I don't know, but they're kind of cool, and I wish we could like actually do the experiments, but for now we can just talk about them. Okay, so this is like a diagram of DNA replication. So remember what I said about how like DNA is anti-parallel to each other, and one side goes from 5 to 3 and the other goes from 3 to 5. So here what we have is, this is one strand of DNA right here. And the helicase unwinds it. And so now it becomes 2 strands that are basically like single strands. This is what RNA would look like this part of it. And we have a leading strand and a lagging strand. And how this works is we have a DNA polymerase, which makes the other part of the DNA. So you can see here how it matches each of these little nucleotides onto the original one, and that makes the DNA sequence again. And this DNA sequence will match the original DNA sequence because you're taking half of it. And this is why it's double-stranded, so that you can like break it apart basically, and then match the original like nucleotides on there, and it'll be the exact same as the parent DNA. So what we have is that this strand is making the DNA from this side to this side, and then it has to go back over here and then do it again, because DNA polymerase only goes one way. I believe it's from 5 to 3, but I'm not sure. So it only goes in this direction. But then this one, it just can flow easily through here because this is like the perfect direction to go in. If you guys take like AP Bio, or maybe even like Bio 1 in like high school, they'll go a lot more into depth about this, but this is kind of just like an overview about it if you're curious. I don't know, feel free to ask me any questions, but yeah, this is a little confusing, I know. Okay. Next, we have cell division. So mitosis is like how cells divide into each other. And we all know that every cell has its own DNA. So how do we get the DNA from one cell to the next cell? And that's what mitosis is. So we have a parent cell right here. And if you guys know, like every cell has, in a human at least, there's 23 chromosomes, sorry. And these chromosomes contain like all your genetic information. So each of these like little line things is like one chromosome, and it's a ton of DNA. Like you might think this is like, oh, that's like one strand of DNA. No, that's like, so it's like a ton of DNA that's condensed into like a linear looking line. And this DNA is then replicated. So it makes these little X shapes. So this line is duplicated so that it makes another one right next to it. And then what happens with these is eventually these two like DNA strands or DNA like segments kind of are like pulled apart. And then it forms two daughter cells, and each of these cells has the exact same DNA as the parent cell. So that is basically the process of mitosis. And then meiosis is, it's like similar too, but meiosis is always used to make like reproductive cells because it only has half the DNA of the original cell. So you need like one from your father and one from your mother, and then that makes like your cells basically. So this is your parent cell, and then the DNA replicates, and it's kind of the same thing that happens here. But instead of like making like each cell, daughter cell have like one of these like chromosomes, like the whole X gets pulled apart. So you start with 4 chromosomes, but here you only have 2 chromosomes, but you have like the same amount of DNA because each of these are duplicated. So this is like what makes it so like your mom and dad like combine their DNA. So some of your chromosomes will come from your mom, some of them come from your dad. And it's obviously not as like simple as that. Like it's not like exactly half, but there's some different like other things that can occur in there. But here we have 2 cells. These are the 2 daughter cells, and they each have half the chromosomes. And then after that, it's kind of the same process as mitosis when these 2 X's get pulled apart and they become the daughter cells. And then these are your like reproductive cells. Yeah. Does anyone have any questions? Okay. Well, then we're going to move on. Okay, so what does DNA actually do? Because right now it's this like weird long chain thing that it can't like perform the functions in your body, right? So what does DNA actually do? DNA, it codes for proteins, and basically your whole body is made of proteins. Like a lot of stuff in your body needs proteins to function. So DNA is replicated over and over again. This is like the process. So DNA is replicated over and over. So you have like many different copies of your DNA. And then after that, it is transcribed. There's a process called transcription right here from DNA to RNA. And what happens in this process is that usually the DNA will split up, and then there'll be an RNA polymerase, which is very similar to DNA polymerase, where it takes nucleotides and binds it to the strand of DNA. But this time, instead of taking DNA nucleotides, it's just RNA nucleotides. So do you guys remember what the difference in the nucleotides between DNA and RNA is? There's only one of them that's different. Okay, remember when, so DNA is ATGC. And then which one, do you guys know which one of those is different in RNA? Like which nucleotide is in RNA that's not in DNA? So that would be U. So U is not in DNA, but it's in RNA and it replaces the T. So there's no T in RNA. So yeah. And then RNA will then be translated into a protein. And usually it uses like RNA, like messenger RNA, and it will like take the amino acids that make up a protein. It'll take the amino acids that match with the 3 RNA nucleotides at a time, and that will make the amino acids, and the amino acids connect into a chain, and then the way that the amino acids bind together makes a protein. Like this whole thing right here is a protein, but notice how it's like a long strand, and the strand is made up of amino acids. These amino acids are slightly bigger than nucleotides, but they're also pretty small, and they interact with hydrogen bonds. Same with DNA. I don't know if I mentioned this earlier, but the nucleotides, the way that they bind to each other is not like actual bonds. It's called something called hydrogen bonds. And that happens when hydrogen atoms interact with other like electronegative atoms. I don't know if you guys know what this is, but yeah, those bind together and it binds them together. And that's the same interactions that protein amino acids have with each other to make proteins. So yeah. The process from RNA to proteins is called translation. Kind of think of it this way, like DNA and RNA are like the same language, just different dialects. So you're just like transcribing it maybe from like transcribe a video into like words. So that's like how DNA is to RNA. But RNA into amino acids, which makes up proteins, is like a completely different language. So you have to like translate the whole thing into a different language. Yeah. That's just a good way to remember it if you ever need that. Okay, now we're going to talk about PCR, which is a laboratory technique that is used to replicate DNA. So say I want to like clone an animal. I mean, I don't think that's possible yet, but okay, say I'm like cloning a bunny and I like take a little piece of some DNA from it, but like I don't want to take too much because like the bunny still needs its DNA. So I take some of its DNA and I want to replicate more of it before I run some like tests on it, like electrophoresis, which is what we're going to learn about later. So what happens is you put it into this machine and you have the original DNA right here. And first we want to separate the 2 strands of it so we can duplicate it. And so the first step is, well, the first step would be to put the DNA in the machine. And then you add like nucleotides and DNA primer, which will help build the rest of the sections of DNA. So then after that, we have denaturation. What happens then is that the DNA is heated up a lot, like almost to like kind of when water boils, like almost that hot usually. And then the hydrogen bonds that connect the different nucleotides will be broken, and you'll have 2 different strands of DNA. And this is called denaturation. And then you have annealing, which takes this DNA primer that only matches to like a specific section of DNA. So say you only want to duplicate a certain section of DNA, this is where you use your DNA primer so that only this section of DNA will be replicated, and then you know which section you have. And then, so in annealing, your sample is usually cooled down a lot so that the DNA primer will bind to the DNA sequence. And then lastly, you have extension. Lastly, you have extension. And during extension, nucleotides are added onto the DNA sequence. And yeah, so that's kind of like the process that DNA polymerase does when it adds different nucleotides to the sample. And this like extends the DNA and will make your replicated DNA strand. So the strand you have right here, this should be the same as this right here. Okay. Has anyone had any experience with PCR? Have you guys heard of it before? Or do you guys have any questions? Okay. Moving on. Next, we have gel electrophoresis. And this is another like lab technique-ish. It is a lab technique. And it is used to separate, it can be used to separate anything from DNA, RNA, proteins, but it's always sorted according to the molecular size. So since, remember we were talking about the structure of DNA, it has phosphate ions on it, which are negatively charged. So in all of DNA, the charge is always proportional to the mass. So like if you have like a section of 3 nucleotides, it'll be a 3- charge. And if you have a section of like 5 nucleotides, it'll be a 5- charge. So the charge and the mass are the same. So basically, gel electrophoresis will separate your DNA sample by the size. So first what happens is the DNA will be cut into different sized fragments, and it'll be cut like using specific specific sequences that you decide. And that's like the cool part, because you can see that like if you put the same amount of DNA into both things, and one of them has like more sections of like a larger size, then you'll know that the DNA doesn't match, because obviously it doesn't have the same DNA sequences as each other. So how this works is you'll have this little box thing basically. And on this side, you have some wells. And in the wells, you're going to put your DNA samples. And usually you like dye your DNA so it's like easier to see. Like it'll be like a different color or something. And what happens is you have this gel here. It can be made of like Argos. I don't know if you guys know what that is, but it's like kind of like gelatin, like a jelly-like substance. And on the side closer to the well, you're going to have a negatively charged battery, and here is a positively charged battery. And once you start the electricity flowing, the more that it moves towards the positive side, the smaller it will be, because it's like, think of it like if I have a sift and then I'm putting things through it, all the small things will fall through, but the big things will stay on top. So that's kind of the same way that the gel works. The bigger pieces of DNA will stay towards the top, and the smaller ones will travel down, down, down. And okay, and then this is kind of what it'll look like. So this is like an end result of like running a gel, as you call it, like running a gel electrophoresis. And the larger pieces of DNA will be at the top, and the smaller pieces will be at the bottom. And this is how you set it up. On the very left, you have a DNA ladder, which is going to have your like some known lengths of DNA. So for example, this might be like 100 base pairs, 200 base pairs, 300 base pairs, 400, 500. And that way you can like match it up. See, like this one lines up with here. So you know that this will have roughly like 200 base pairs and this one will have like 100 base pairs. So like, base pairs are like your nucleotides. So how many like A's, T's, G's, C's there are. And yeah, so the DNA ladder is usually on this very side, and then you can compare two different DNA samples to see if they line up or not. And this just helps you like determine the sequence of a genome sequence. And basically like all humans are like 99 point like, I don't know, 99 point something percent DNA is similar. So like you're gonna have a lot of the same things. So you have to look at those sequences that are different. And that's gonna tell you like who this DNA may implicate or something like that. So yeah, if you're always gonna have some that line up. So then that just like tells you that it's like working or something like that. So also, like PCR, which we talked about earlier, like you guys have probably heard of like a PCR COVID test, because that's like very relevant to what we're doing right now. And what that does is it like takes the cells, usually you do like a nose swab or something, and it'll take your like cells and it'll replicate it and it'll like look for the sequences that have that COVID gene in it, because COVID is like a virus. So it basically like inserts its DNA into you. And so once you do a PCR test, it looks for those gene sequences and if they're in your cells or not. And that's how it like detects if you have COVID or not. So yeah, that's pretty cool.
I thought the COVID vaccine, like, doesn't the COVID vaccine also show them what like the COVID virus looks like? And then so isn't that what a vaccine does though?
Yes. So when you get a vaccine, it does also inject like the virus into you, but usually in a vaccine, it's not going to have like it's like a modified version. So it's not going to have like the infectious part, like the genome sequence of the virus. And also, I'm pretty sure when they're like detecting the gene sequence in like when you like get a COVID test, it has to be like a certain amount. Like I'm pretty sure like if you have like actually have COVID, it'll be more than if you just have the vaccine. And also I think they would look for different sequences than the one in like the vaccine, or else they would not be able to tell if you had it or not. So yeah, that's kind of how that works. But yeah, that's really cool that like how PCR works and how like it can actually detect if you have a disease. Okay, so this is kind of what it looks like when you perform gel electrophoresis. This one is like first you have the markers, and then so here's like how many base pairs would be on this side. So you like know that this has like 1,200 base pairs or something like that. And then A, B, C, D. It's like the different DNA samples, and it'll just like line up here, and then you know this has like 1,000 base pairs in it, and then this is like, yeah, so on. This is like a cooler looking one. It's like glowing, and this is another DNA sample. Yeah, so if you can see like this matches up, it could say that these two DNA sequences share some sequence in the middle of it or something like that. Okay, so this is like an example of like, I guess like your crime scene. So for example, you find this DNA on your crime scene and you run it through a gel electrophoresis, and this is what comes out of it. And then you have suspect 1, 2, 3. So obviously you can see that 2 matches the crime scene exactly, but this isn't gonna always happen. Like you're not just gonna like find DNA on your crime scene and match it to your person and it like matches perfectly, but it'll look somewhat like this. And you can see that 1 is kind of similar, but it's like a little bit different here, and then 3 is like different down here. So that's like what it'll look like when you're like comparing your different gel electrophoresis sequences. And gel electrophoresis is kind of cool too. Like you can do it at home. I did like this experiment thing kind of last year at home where you just take some gel, like this like Argos gel, and then you have batteries on each side, and then you can put different like food colorings in, and it'll actually separate out the food coloring by the size of the molecules in the food coloring. And yeah, so it's like not a super complicated like lab thing, but it's really useful in all different like methods of science. And then here's some information to know for like, I don't know. So CODIS is the Combined DNA Index System. And this is basically where the FBI keeps like all the DNA sequences of like criminals and stuff. And you and me, our DNA is probably not in the system, just because we probably have not been arrested for any crime or any like major crime. And because otherwise they will not have your DNA. Like when you're born, they do not take your DNA, and they don't just like randomly go around taking your DNA. So this is usually only for like previous offenders. So sometimes when they find like DNA at a crime scene and they check it, run it through the system, and maybe this person has committed a crime before and been in jail before, and then they'll find a match to the DNA sequence. But most of the time they do not. And this is the same thing with fingerprints. So yeah. Here's some more information in this link if you want it about CODIS and like how they really analyze the DNA and like what their system is like. Yeah, the FBI website is actually really cool. There's some really cool things on there.